Welcome to Ultimate Sleep Doctor. Tonight, we'll journey into the quiet wonders of our own being. Consider the intricate dance of life, the way our bodies and minds are built from the very building blocks of our existence. We explore gene therapy, a field where science gently rewrites the code of our health, a subtle conversation with the universe within. It's a fascinating glimpse into the potential for healing, a reflection on the profound interconnectedness of all things. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe. Let your thoughts drift like clouds across a tranquil sky. Let's begin. The dream of correcting faulty genes has a surprisingly long and winding history, stretching back much further than you might think. It's not a story that began with gleaming laboratories and sophisticated technology. Instead, it started with curiosity, observation, and a persistent, almost magical belief that the very blueprints of life could be changed. The earliest whispers of gene therapy emerged from the study of infectious diseases, particularly in the late 19th and early 20th centuries. Scientists were just beginning to understand that diseases weren't always caused by harmful substances, like bacteria or toxins, but could also arise from disruptions within the cells themselves. A pivotal figure in this early exploration was Wilhelm Behring, a German physician who, along with Emil von Behring, developed serum therapy for diphtheria in the 1890s. This groundbreaking work demonstrated that antibodies, specialized proteins produced by the immune system, could neutralize toxins, essentially fixing the problem at a molecular level. This was a crucial step, as it established the idea that a foreign agent could be used to correct a defect in the body. It wasn't gene therapy as we understand it today, of course, but it planted the seed of the concept. Could we deliver something to a cell to restore its function? Around the same time, Russian scientist Dmitry Ivanovsky was studying tobacco mosaic virus, a virus that caused a disease in tobacco plants. Ivanovsky, along with Barbara McClintock, observed that the virus didn't destroy the plant cells, but instead seemed to be incorporated into them. McClintock, in particular, made a remarkable discovery. She noticed that corn plants infected with the virus displayed unusual chromosomal activity, as if the virus was rearranging the genes within the cell. McClintock's work, initially met with skepticism, would later be recognized as a fundamental contribution to the understanding of genetic inheritance and transposition, the ability of genes to move around within the genome. Her observations, though not immediately applied to human medicine, hinted at the possibility of harnessing viruses to interact with cells in a controlled manner. The next significant development arrived in the 1960s, largely thanks to the work of James Edward Fruchtwiller and David Russell at the University of Nebraska Medical Center. They were studying the effects of adenoviral vector therapy, a treatment for spinal muscular atrophy, SMA, a devastating genetic disease that causes muscle weakness and atrophy. SMA is caused by a mutation in the SMN1 gene, and the researchers noticed that adeno-associated virus, AAV, A common virus found in many animals could deliver a functional copy of the gene to cells in the spinal cord of mice. What's truly fascinating is that Frutwiller and Russell hadn't initially set out to create a gene therapy treatment. They were investigating the effects of AAV on cellular growth, but they stumbled upon the incredible ability of the virus to deliver genetic material. They used AAV because it was relatively safe and easy to work with, and it could efficiently infect cells. This accidental discovery was the first documented instance of a virus being used to deliver a therapeutic gene to cells, a pivotal moment in the history of gene therapy. However, the initial attempts faced serious challenges. The immune system reacted strongly to the AAV, triggering an inflammatory response that limited the amount of gene delivered and often caused harm. This was a common problem with early viral vectors. The body's defenses recognized the virus as a threat and attacked it, neutralizing the therapeutic effect. Scientists realized they needed to find ways to cloak the viral vector, shielding it from the immune system and allowing it to safely deliver its genetic payload. As research continued, scientists began to explore other viruses, including herpes simplex virus HSV and retroviruses like HIV. These viruses had characteristics that made them potentially better candidates for gene therapy vectors. HSV, for instance, could be engineered to remove genes that would trigger an immune response, and retroviruses, while known for their ability to integrate permanently into the host genome, could be manipulated to deliver genes without causing uncontrolled cell division, a major concern with earlier retroviral vectors. The focus shifted to understanding how to control the integration process and minimize the risk of causing cancer. The development of adenoviral vectors, initially discovered almost by accident, laid the foundation for much of the subsequent research. Scientists began to investigate methods for modifying the virus, such as removing genes that would provoke an immune reaction. They also started to explore ways to target specific cell types, improving the precision of gene delivery. This early work, building upon the observations of Ivanovsky, McClintock, and Frutwiller, marked the beginning of a journey that would ultimately transform our ability to treat diseases at the genetic level. The pursuit of controlling viruses for therapeutic purposes, once a largely accidental exploration, was rapidly becoming a deliberate and increasingly sophisticated endeavor. Let's delve into the heart of what makes gene therapy so fascinating: the intricate relationship between our genes and how they cause illness. It all begins with understanding mutations, tiny changes in our DNA sequence. DNA, you see, is like a very long instruction manual for building and running our bodies. These instructions are written as a series of letters: A, T, C, and G. The order of these letters dictates everything from eye color to how our cells function. Most of the time, this sequence is perfect and everything runs smoothly. But sometimes, errors occur during DNA copying, or damage from things like radiation or certain chemicals can introduce these mutations. These errors aren't always harmful. Occasionally, they don't have any noticeable effect. However, in some cases, a mutation can disrupt the precise instructions, leading to a disease. Now, let's consider a specific example: cystic fibrosis. Cystic fibrosis is a genetic disorder that primarily affects the lungs and digestive system. It's caused by a mutation in a gene called CFTR, the cystic fibrosis transmembrane conductance regulator. This gene normally produces a protein that acts like a gatekeeper, controlling the flow of salt and water across cell membranes. Think of it like a tiny faucet that regulates the amount of water coming in and out of the cells, lining your airways and intestines. The CFTR protein is crucial for keeping these membranes healthy and functioning correctly. When the CFTR protein isn't working properly, because of the mutation, the cell membranes become abnormally thick and sticky. In the lungs, this leads to the buildup of mucus, which clogs the airways, making it difficult to breathe and increasing the risk of infection. In the digestive system, it interferes with the absorption of nutrients from food. The most common mutation in CFTR, responsible for approximately 70% of cystic fibrosis cases, is known as the delta F508 mutation. This particular mutation causes the CFTR protein to be largely destroyed before it can even reach the cell membrane. It's like the protein is immediately broken down, preventing it from doing its job. There are other, rarer mutations that also disrupt CFTR function, but delta F508 is the most frequent. The severity of cystic fibrosis depends on the type and number of mutations a person carries. Someone with 2 copies of the delta F508 mutation will typically have the most severe form of the disease. Individuals with 1 copy are often referred to as carriers, and they usually don't experience any symptoms themselves. However, they can pass the mutated gene to their children. Scientists have spent years trying to understand precisely how these CFTR mutations impact the cell's ability to regulate salt and water. Research has revealed that the mutations often result in the CFTR protein being misfolded, meaning it doesn't fold correctly into its proper 3-dimensional shape. This misfolded protein cannot perform its function properly, and it's often targeted for destruction by the cell's own cleaning mechanisms. Furthermore, the mutations can affect the way the CFTR protein interacts with other proteins involved in its transport to the cell membrane. These interactions are critical for the protein to reach its destination and function effectively. It's a complex chain of events, and the exact details are still being investigated. Gene therapy for cystic fibrosis aims to address these problems. The basic idea is to introduce a healthy copy of the CFTR gene into the patient's cells. There are different approaches to achieving this. One method involves using a modified virus, a very safe, harmless version, to deliver the functional CFTR gene into the patient's lung cells. The virus acts as a delivery vehicle, carrying the gene into the cells. Another approach involves using a technique called CRISPR-Cas9 gene editing. CRISPR-Cas9 is a revolutionary technology that allows scientists to precisely cut and paste DNA sequences. In the case of cystic fibrosis, CRISPR-Cas9 could potentially correct the mutated CFTR gene directly, restoring its normal function. It's a very promising tool, though it's still being developed and refined. The goal of gene therapy in cystic fibrosis isn't necessarily a permanent cure, although that's the ultimate aspiration, but rather to improve the function of the CFTR protein and reduce the symptoms of the disease. Clinical trials are underway, and early results have been encouraging, showing improvements in lung function and a decrease in mucus buildup in some patients. It's important to remember that gene therapy is a relatively new field, and there are still many challenges to overcome. The long-term effects of gene therapy are not fully known, and there are potential risks, such as immune reactions. However, the progress made in understanding the genetic basis of cystic fibrosis and the development of gene therapy approaches represent a significant step forward in the fight against this debilitating disease. The intricate interplay between genes and disease, as exemplified by cystic fibrosis, highlights the remarkable complexity of our bodies and the potential of gene therapy to target the root cause of illness. It's a story of scientific discovery, hope, and the ongoing pursuit of better health for all. The quest to deliver therapeutic genes directly into human cells has always relied on a clever trick, harnessing the natural abilities of viruses. Early attempts, using retroviruses and lentiviruses, were groundbreaking, but they came with significant challenges, like the potential for the virus to integrate permanently into the host's genome, sometimes causing unwanted side effects. Scientists needed a gentler, more controllable tool, one that wouldn't dramatically alter the cell's own DNA. This is where the adeno-associated viruses, or AAVs, stepped into the spotlight. AAVs are remarkably well-behaved viruses. Unlike retroviruses, they don't integrate into the host's DNA. Instead, they establish a temporary, borrowed relationship with the cell. They're naturally found in humans and other primates, and they've evolved a special partnership with the immune system. AAVs aren't particularly harmful. The body recognizes them as harmless, and rather than attacking them, it actually helps them deliver their genetic cargo. This inherent safety profile is a crucial reason for their popularity in gene therapy. The secret to AAV's success lies in its simple life cycle. AAVs are tiny, almost spherical viruses, and they're built for efficiency. Once they enter a cell, they release their genetic payload, the therapeutic gene, into the cell's nucleus. But that's where the story truly begins. The AAV itself doesn't stay around. It doesn't replicate, it doesn't cause any lasting changes. It simply exits the cell, leaving the therapeutic gene behind. Now, the gene itself needs to get to work. This is where the careful engineering of AAVs comes into play. Researchers have learned to modify the AAV capsid, the protein shell that surrounds the genetic material, to improve its targeting abilities. Think of the capsid like a delivery truck. The original trucks were pretty good, but scientists figured out how to add GPS systems and specialized routes to ensure the cargo arrived precisely where it was needed. Different AAV serotypes, these are essentially different versions of the virus, have a natural affinity for different cell types. Some AAVs are particularly good at targeting liver cells, which is why they're commonly used in liver-targeted therapies. Others are designed to target muscle cells, neurons, or even specific immune cells. The researchers select the AAV serotype that best matches the intended target, maximizing the chance that the therapeutic gene will reach the right cell. This targeting isn't just about getting to the right cell. It's about controlling how the gene is expressed. The amount of protein produced by the newly delivered gene can be carefully regulated. Scientists can engineer AAVs to express the therapeutic gene for a short period, just long enough to make a beneficial effect, or to express it continuously, depending on the disease being treated. Furthermore, the capsid itself can be decorated with antibodies, proteins that specifically recognize and bind to certain cell types. This technique, called antibody-mediated targeting, adds an extra layer of precision to the delivery process. It's like adding a special signpost to the delivery truck, guaranteeing it stops only at the intended address. The beauty of AAVs is also reflected in their ability to cross the blood-brain barrier, a notoriously difficult obstacle for many gene therapies. While still an area of active research, certain AAV serotypes have demonstrated the capacity to effectively deliver genes to neurons in the brain, opening up possibilities for treating neurological disorders. The development of AAV vectors isn't a simple, one-size-fits-all approach. There's a significant amount of research dedicated to optimizing AAV vectors for specific applications. This involves understanding the intricate interactions between the AAV capsid, the therapeutic gene, and the host cell's immune system. Scientists are constantly refining the design and production of AAV vectors, aiming to improve their efficiency, safety, and targeting capabilities. Beyond simply delivering genes, AAV vectors are being explored for other applications, such as gene editing. Researchers are developing AAV-based systems that can deliver CRISPR-Cas9 enzymes, allowing for precise modifications of the genome. This opens up even more possibilities for treating genetic diseases. The journey of AAVs in gene therapy is still ongoing, but their inherent safety and versatility have made them a cornerstone of this rapidly advancing field. Ongoing research is focused on overcoming challenges, such as immune responses to the AAV capsid, and ensuring long-term gene expression. With each new discovery, AAVs continue to play a vital role in the promise of treating diseases at their genetic root. Looking ahead, the future of AAV-based gene therapy appears bright. Scientists are exploring new AAV serotypes, developing innovative targeting strategies, and refining manufacturing techniques. The continued exploration of this technology promises to unlock even greater potential for treating a wide range of diseases, offering hope and new possibilities for patients worldwide. The world of gene therapy took a truly remarkable leap forward with the development of CRISPR-Cas9, a technology that felt almost like magic to scientists. Before CRISPR, editing genes was a painstakingly slow, complex, and often unreliable process. It involved introducing entirely new genes or trying to carefully replace faulty ones, a process that frequently led to unintended consequences within the cell. CRISPR-Cas9 changed everything, offering a far more precise and efficient method for targeting and modifying DNA. At its heart, CRISPR-Cas9 is based on a system naturally found in bacteria. These bacteria use a protein called Cas9. Think of it as a tiny molecular scissor to defend themselves against viruses. When a virus attacks, the bacteria recognize it and use Cas9 to cut the viral DNA, effectively neutralizing the threat. Scientists realized that this bacterial defense system could be adapted for a much more controlled purpose: to edit our own DNA. The system works like this. CRISPR, short for clustered regularly interspaced short palindromic repeats, is actually a guide. It's a small piece of RNA, a molecule that carries genetic information, designed to match a specific sequence of DNA within our genome. This guide RNA is carefully crafted to be complementary to the target DNA sequence we want to modify. It's like a GPS, directing the Cas9 protein to the exact location on the DNA strand. The Cas9 protein is the workhorse of the system. Once the guide RNA has successfully located its target sequence, Cas9 makes a precise cut in the DNA. This isn't a random cut. It's a carefully targeted break, just where the guide RNA directed it. The cell recognizes that this DNA has been damaged and triggers its natural repair mechanisms. There are two main ways the cell responds to this DNA break. The first is called non-homologous end joining, NHEJ. This is a quick and dirty repair pathway, often resulting in small insertions or deletions of DNA bases, like removing a few building blocks from a Lego creation. While this can be useful for disrupting a gene entirely, making it unable to function, it's also prone to errors. The second and far more precise pathway is called homology-directed repair, HDR. If scientists provide the cell with a DNA template, a perfect copy of the desired DNA sequence, the cell can use this template to repair the break, effectively inserting the new sequence into the genome. This is like providing the cell with the correct instructions to rebuild a section of the Lego creation exactly as intended. HDR offers much greater control over the outcome of the gene editing process. The initial applications of CRISPR-Cas9 were incredibly exciting. Researchers quickly began exploring its potential in treating a wide range of diseases. In labs around the world, scientists were using CRISPR-Cas9 to correct genetic mutations in human cells, first in Petri dishes, and then in animal models. One of the earliest and most promising applications focused on sickle cell anemia and beta thalassemia, both caused by single-base mutations in the beta-globin gene. CRISPR-Cas9 allowed researchers to directly correct these mutations, restoring the ability of the cells to produce normal hemoglobin. In animal models, where scientists used CRISPR-Cas9 to edit the genes responsible for these blood disorders, they observed a significant improvement in the animal's health, demonstrating the transformative potential of the technology. Beyond blood disorders, CRISPR-Cas9 was investigated for its potential in treating inherited eye diseases like Leber congenital amaurosis, a genetic blindness caused by mutations in the CEP290 gene. Researchers successfully used CRISPR-Cas9 to introduce a corrected copy of the gene into the retinal cells of mice, leading to improved vision. Another area of intense research involved cancer treatment. Scientists explored using CRISPR-Cas9 to engineer immune cells, like T cells, to specifically target and destroy cancer cells. By modifying the T cells to recognize unique markers on the surface of tumor cells, CRISPR-Cas9 offered a potential approach to immunotherapy. The technology's rapid advancement wasn't without challenges. Ensuring that the CRISPR-Cas9 system only targeted the intended DNA sequence, avoiding off-target effects where it cuts at unintended locations, was a major concern. Scientists developed various strategies to improve the specificity of the system, including designing more accurate guide RNAs and using modified Cas9 proteins. Furthermore, delivering the CRISPR-Cas9 components, the guide RNA and the Cas9 protein, to the correct cells within the body posed a significant hurdle. Different delivery methods were investigated, including viral vectors, similar to those used in traditional gene therapy, and lipid nanoparticles. The ongoing research continues to refine these delivery methods, making them safer and more effective. Despite these challenges, CRISPR-Cas9 represents a monumental shift in our ability to manipulate the human genome. It's a powerful tool with the potential to revolutionize medicine, offering hope for treating diseases that were previously considered incurable. The journey of CRISPR-Cas9 is still unfolding, but its initial applications and promise have undeniably ushered in a new era of genetic research and treatment. The quest to harness the power of genes for healing takes many fascinating routes, and a critical distinction in this journey revolves around where the therapeutic gene is introduced into the body. We've seen how viruses like AAVs can carry genetic material, but the precise strategy, whether the initial work happens outside the patient's body and then delivered, or directly within, dramatically impacts the process. These approaches are broadly categorized as ex vivo and in vivo gene therapy, and understanding their differences is key to appreciating the complexities of this evolving field. The ex vivo strategy, meaning outside the body, begins with a small sample of the patient's cells. Typically, this might be bone marrow stem cells, blood cells, or even skin cells. These cells are carefully harvested, often through a minimally invasive procedure. Once in the lab, these cells become the focus of intense genetic modification. Scientists use techniques like viral vectors, similar to those we've discussed, to introduce the corrected gene into the cells. The modified cells then grow and multiply in a controlled environment, transforming into a healthy, gene-corrected population. This carefully crafted cell population is then returned to the patient, usually through a single infusion. The beauty of ex vivo gene therapy lies in the meticulous control. Scientists can select for the best corrected cells, minimizing the risk of unwanted side effects. Furthermore, the environment within the lab provides an ideal setting for the modified cells to thrive and produce the therapeutic protein. This approach is particularly useful when targeting specific cell types, ensuring that the corrected gene is delivered precisely where it's needed. gene therapy for severe combined immunodeficiency, SCID, often called bubble boy disease, has historically relied heavily on an ex vivo approach, where bone marrow stem cells are modified and then reintroduced to the patient. Now, let's shift our perspective. In vivo gene therapy represents a different approach, a direct delivery of the therapeutic gene directly into the patient's body. This method bypasses the need to isolate and modify cells outside the body, offering a potentially simpler and more convenient treatment option. The challenge with in vivo gene therapy is controlling where the gene goes and how it's expressed. Delivery methods are incredibly varied. Researchers are exploring several techniques, including direct injection of viral vectors, lipid nanoparticles, and even modified bacteria. Lipid nanoparticles, for example, are tiny bubbles made of fat molecules that can encapsulate the gene and deliver it to specific tissues. Scientists are working to modify these nanoparticles to target specific cell types, enhancing their precision. Another strategy involves using modified bacteria, which can be engineered to carry the therapeutic gene and deliver it directly to the affected area. However, ensuring these bacteria don't cause any unintended consequences is a significant hurdle. The success of in vivo gene therapy hinges on the efficiency of gene delivery and the stability of the introduced gene. The body's immune system can recognize and destroy viral vectors or nanoparticles, reducing the effectiveness of the treatment. Researchers are constantly developing strategies to evade the immune response, such as using attenuated, weakened viruses or engineering vectors with cloaking properties that make them less visible to the immune system. Another consideration is the duration of gene expression. Ideally, the introduced gene should produce the therapeutic protein for an extended period. Scientists are investigating ways to improve the stability of the gene within the cell's DNA, potentially through techniques like integrating the gene into the host's genome, though this comes with its own set of potential risks. Comparing the two approaches reveals distinct strengths. Ex vivo gene therapy offers greater control over the modified cells and reduces the risk of systemic side effects. It's particularly well suited for diseases where targeting a specific cell type is paramount. However, it requires multiple procedures, the cell harvest and return, which can be burdensome for patients. In vivo gene therapy, on the other hand, is potentially simpler, offering a single treatment. It's also more adaptable to diseases affecting multiple tissues. But achieving precise targeting and controlling long-term gene expression remain significant challenges. Furthermore, the choice between ex vivo and in vivo isn't always a strict dichotomy. Researchers are increasingly exploring hybrid approaches that combine the benefits of both. For instance, a patient's cells might be modified ex vivo and then administered in vivo. The long-term goal is to refine these techniques, creating customized gene therapies tailored to the individual patient's needs. Each approach carries inherent risks and complexities, and ongoing research is focused on improving safety, efficacy, and delivery methods. The journey of gene therapy is still unfolding, and the interplay between ex vivo and in vivo strategies will undoubtedly shape its future. Ultimately, The ability to manipulate and harness the power of genes holds immense promise for treating a wide range of diseases, and understanding the nuances of these delivery methods is central to realizing that potential. The careful delivery of therapeutic genes into a patient's body is a wonderfully complex dance. We've explored how vectors, often viruses cleverly modified to be safe, carry these genes, and how different methods, like treating cells outside the body and then returning them, or directly injecting the gene, can bring about change. But this intricate process isn't without its hurdles. Perhaps the biggest and most fascinating of these hurdles resides in the body's own defense system, the immune system. When a viral vector is introduced, the body doesn't simply welcome the new genetic instructions, it reacts. This reaction, a series of immune responses, can dramatically affect the success of gene therapy. Viral vectors, even the most meticulously engineered ones, still possess recognizable components that trigger immune responses. These components, proteins from the virus itself, and sometimes fragments of the delivered gene, are identified as foreign by the immune system. This initial recognition starts with innate immune cells, like macrophages and dendritic cells. These cells are constantly patrolling the body, scanning for anything that shouldn't be there. Upon encountering viral proteins, they become activated, releasing signals that alert the rest of the immune system. This activation leads to the amplification of the response. Dendritic cells, particularly, are crucial. They eat the viral vector or its components and then travel to lymph nodes, presenting these foreign antigens, small pieces of the vector, to T cells. T cells are the commanders of the adaptive immune system, recognizing specific antigens and orchestrating a targeted attack. There are different types of T cells involved. Helper T cells coordinate the response, boosting the activity of other immune cells. Cytotoxic T cells, on the other hand, directly attack cells displaying the viral antigens on their surfaces, attempting to eliminate them. Now, the type of viral vector used plays a significant role in the strength and nature of this immune response. Adeno-associated viruses, AAVs, which we've previously discussed, are generally considered to elicit a weaker immune response compared to some other vectors like lentiviruses. This is largely due to AAVs' inherent characteristics. They don't actively replicate within cells, and their capsid, the outer shell, is less likely to trigger strong inflammatory signals. However, even AAVs can induce an immune response, especially with repeated administrations. The capsid itself contains many proteins that the immune system can recognize. Furthermore, the dosage, the amount of viral vector delivered, is a critical factor. Higher doses can overwhelm the immune system, leading to a more pronounced and potentially detrimental response. It's a delicate balancing act. Too little vector, and the gene isn't delivered effectively. Too much, and the immune system kicks into overdrive. Researchers carefully assess the vector's concentration to optimize therapeutic efficacy while minimizing immune complications. A particularly concerning phenomenon is the development of neutralizing antibodies. As the immune system encounters the viral vector, it starts producing antibodies specifically designed to bind to the capsid proteins. These antibodies aren't necessarily harmful on their own. However, they can effectively block the vector from infecting the cells. If the vector can't enter the cell to deliver the therapeutic gene, the gene therapy simply fails. This can also lead to the vector being cleared from the body more quickly, further reducing its chances of success. Beyond neutralizing antibodies, there's evidence suggesting that the immune system can initiate a more profound inflammatory reaction. The presence of viral proteins and the subsequent activation of immune cells can trigger the release of inflammatory cytokines, chemical messengers that promote inflammation. Excessive inflammation can damage surrounding tissues and hinder the gene's intended function. It can also prevent the new gene from being properly expressed within the cell. Researchers are actively exploring strategies to mitigate these immune responses. One approach involves using immunosuppressive drugs to dampen the immune system's activity. However, this carries the risk of increasing the patient's susceptibility to infections. Another strategy focuses on evading the immune system. Scientists are engineering vectors with modified capsids and changing the proteins on the outside to make them less recognizable to the immune system. This can involve deleting certain capsid proteins or introducing new ones that are less likely to trigger an immune response. Another avenue of research involves pulse delivery, delivering the gene therapy in a carefully controlled manner, perhaps intermittently, to avoid constant immune stimulation. The goal is to allow the immune system to adapt and tolerate the vector over time. There is also work on immune suppression at the cellular level, attempting to alter the gene expression patterns within the cells themselves to reduce the inflammatory signals. The challenge, ultimately, lies in achieving a delicate balance. We need to deliver therapeutic genes effectively, while simultaneously preventing the immune system from aggressively rejecting the vector. It's a complex, ongoing area of research, and advancements in this field are crucial for the continued development and success of gene therapy as a treatment option for a wide range of diseases. Understanding and controlling these immune responses represents a pivotal step towards harnessing the full potential of this transformative technology. The steady hum of the lab felt almost comforting, a quiet counterpoint to the incredible activity unfolding within. For years, gene therapy had largely relied on simply delivering a corrected version of a faulty gene, a blueprint for the cell to rebuild itself. But scientists realized that this was often too simplistic, too blunt. Cells, particularly within complex organisms, don't operate with a single switch. They respond to intricate networks of signals, feedback loops, and carefully timed interactions. This led to a fascinating new direction: the design and construction of synthetic gene circuits. These weren't about replacing a broken gene, but about programming cells to behave in a specific, controlled manner, like tiny biological computers. The foundational idea stemmed from the observation that natural cells already possessed remarkably sophisticated regulatory systems. Cells respond to everything from nutrient availability and light exposure to the presence of other cells. Scientists began to mimic these natural regulatory mechanisms, building circuits out of DNA sequences that interacted with each other in predictable ways. A typical circuit might consist of logic gates, DNA sequences that act like or, and not gates in electronics. An AND gate, for example, requires multiple input signals to be present before triggering an output. A simple circuit might use this to ensure that a therapeutic protein is only produced when a particular cell type is present, preventing unwanted side effects in other tissues. Early synthetic circuits were constructed using relatively straightforward DNA sequences. Researchers would meticulously design sequences that responded to specific stimuli, often using promoters, DNA regions that control the expression of genes, as the switches. One pioneering approach utilized bacterial repressors, naturally occurring DNA sequences that limit gene expression. Scientists adapted these repressors to respond to human-specific signals, effectively creating miniature control panels within cells. These circuits could be programmed to activate or inhibit gene expression based on external cues, like a specific chemical or a change in pH. The beauty of this approach wasn't just in the individual components, it was in the way they were combined. Complex circuits could be built by linking multiple logic gates together, creating cascading effects. For instance, a circuit might be designed to respond to inflammation. If inflammatory molecules are detected, the initial trigger sequence activates a second sequence, which in turn activates the production of a protein that suppresses the immune response. This targeted response is far more precise than simply shutting off an entire gene, potentially minimizing collateral damage. As the technology matured, scientists began incorporating entirely synthetic DNA sequences that didn't exist naturally in living organisms. This opened up a huge range of possibilities, allowing for the creation of circuits with unprecedented complexity and sensitivity. Researchers started using computational modeling to design these circuits, simulating their behavior before actually building them in cells. This allowed them to optimize the circuits for maximum effectiveness and minimize the risk of unintended consequences. Imagine designing a circuit to detect and destroy cancer cells, a circuit that would only activate when specific markers found on the cancer cell surface were present, sparing healthy cells from harm. Another significant advancement was the development of tunable gene circuits. These circuits could be dynamically adjusted, allowing researchers to fine-tune the response of the cell. This was achieved by incorporating elements that could be modified. For example, using a system that could respond to different levels of a particular signal. This adaptability is crucial for treating diseases that fluctuate over time, such as diabetes, where blood sugar levels vary depending on diet and activity. Furthermore, the development of sophisticated delivery methods became intrinsically linked to the design of synthetic circuits. If a circuit needs to operate in a specific location within the body, the delivery system needs to be able to precisely target that location. Researchers are now exploring ways to incorporate the circuit directly into the delivery vehicle, creating a self-contained therapeutic system. This approach reduces the risk of immune responses and ensures that the circuit operates as intended. The potential applications of synthetic gene circuits are vast. Beyond cancer therapy, they could be used to treat neurological disorders, cardiovascular diseases, and infectious diseases. Imagine a circuit that could repair damaged neurons after a stroke, or one that could control the release of insulin in response to changes in blood glucose levels. The field is still relatively young, but the early results are incredibly promising. Current research focuses on creating circuits that are not only effective, but also safe and predictable. Scientists are working on developing methods for rigorously testing these circuits in vitro, in lab settings, and in vivo, in living organisms, to assess their long-term stability and potential side effects. The ultimate goal is to create truly personalized therapies, tailored to the specific needs of each patient. The complex, beautiful world of synthetic gene circuits represents a fundamental shift in how we approach gene therapy, moving beyond simple gene replacement to a future of precisely controlled, intelligent biological systems within our cells. The world of genetic medicine holds remarkable potential to address some of the most challenging diseases, and one area experiencing particularly exciting advancements is spinal muscular atrophy, or SMA. It's a condition that profoundly impacts muscle strength and movement, primarily caused by a missing or malfunctioning gene called SMN1. This gene is crucial for producing proteins that protect nerve cells, specifically motor neurons, vital for controlling muscle movement. Without enough of these protective proteins, motor neurons begin to waste away, leading to progressive muscle weakness and eventual paralysis. The severity of SMA varies greatly, and current treatments primarily focus on managing the symptoms, offering a life of several years for many affected individuals. However, gene therapy represents a truly transformative approach, aiming to correct the underlying genetic defect. Several innovative clinical trials are now underway, each utilizing different strategies to deliver a functional copy of the SMN1 gene into patients' bodies. One of the most promising approaches involves ex vivo gene therapy, where a patient's own cells are taken, genetically modified in a laboratory setting, and then returned to the patient. In a trial led by Eversana, scientists extracted bone marrow stem cells from children with SMA. These cells were then engineered to carry a healthy copy of the SMN1 gene using an adeno-associated virus, AAV, as the delivery vehicle. This AAV, a common and relatively safe viral vector, acted like a tiny courier, safely transporting the gene into the stem cells. The modified stem cells were then infused back into the children. Results from this early trial have been truly remarkable, demonstrating significant improvements in motor function and muscle strength in several participants. Some children who were previously unable to sit independently are now able to do so, and others have shown increased mobility changes that were previously considered unattainable. Another avenue of research is focused on in vivo gene therapy, aiming to deliver the SMN1 gene directly into the patient's nervous system. This is a more complex undertaking than ex vivo approaches, primarily because delivering gene therapy agents to the central nervous system is notoriously difficult. A significant effort is directed at developing AAV vectors optimized for crossing the blood-brain barrier, the protective membrane that shields the brain from harmful substances. Scientists are constantly refining these vectors, making them more efficient at reaching motor neurons. A trial run by Vertex Pharmaceuticals and Biogen is specifically targeting this challenge. They are using an AAV vector designed to deliver the SMN1 gene directly to motor neurons within the spinal cord. This trial is investigating whether sustained expression of the SMN1 gene, achieved through this targeted delivery, can effectively halt or slow down the progression of SMA. Beyond simply delivering a functional copy of the gene, researchers are exploring ways to boost the levels of SMN protein produced. A key insight is that SMA patients often have reduced levels of SMN2, a related gene that can partially compensate for the missing SMN1. The goal is to enhance the production of SMN2. Trials are evaluating the use of small molecules that increase the efficiency of SMN2 transcription. The process of copying the SMN2 gene into RNA, which then becomes protein. Preliminary results from these studies are generating considerable optimism, suggesting that bolstering SMN2 expression can dramatically improve patient outcomes. Furthermore, scientists are investigating the use of CRISPR-Cas9 gene editing technology, another powerful tool in genetic medicine. While still in early stages of development for SMA, CRISPR-Cas9 could potentially be used to directly correct the mutation in the SMN1 gene itself. This approach, known as gene correction, would be significantly more precise than simply delivering a functional copy of the gene. However, delivering CRISPR-Cas9 to motor neurons remains a formidable challenge, and the technology's safety and efficacy are still being rigorously assessed. The clinical trials involving SMA are representing a significant leap forward in gene therapy. The data emerging from these studies is fueling hope for individuals and families affected by this devastating disease. While challenges remain, including the long-term durability of the gene therapy effects and the potential for immune responses, the progress achieved is truly remarkable. Researchers are continually refining their strategies, developing more effective delivery methods, and exploring novel therapeutic approaches. The ultimate aim is to develop a cure for SMA, offering a future where children born with this condition can live full and active lives. Ongoing monitoring and advancements in gene therapy technologies are poised to transform the landscape of SMA treatment, paving the way for a new era of personalized medicine. The whispers about gene therapy have grown louder, haven't they? We've seen how carefully engineered viruses can deliver new instructions to cells, how complex circuits can be built within genes to control their activity, and the remarkable progress being made in treating diseases like spinal muscular atrophy. But beneath all this exciting medical potential lies a profoundly complex question, one that forces us to grapple with the very nature of inheritance and what it means to alter the human story. This question centers around something called germline gene therapy, and it's a territory where the stakes are unlike anything we've encountered before. Germline gene therapy, in essence, isn't about fixing a single illness in a living person. It's about making changes to the DNA within sperm or eggs, or even very early-stage embryos. These changes wouldn't just affect the person receiving the treatment. They would be passed down to all subsequent generations. It's like planting a seed that grows into a tree, and that tree produces fruit, in this case, fruit that carries the modified DNA. The implications are staggering. If successful, germline gene therapy could, theoretically, eliminate inherited diseases from a family's lineage altogether. Imagine a future where cystic fibrosis, Huntington's disease, or sickle cell anemia simply disappear because the faulty gene was corrected in the very beginning. However, this seemingly utopian vision quickly reveals a tangled web of ethical concerns. The first, and perhaps most immediate, is the question of consent. An embryo or a very young child, who is the recipient of a germline modification, cannot consent to that change. They have no voice in the decision, no ability to understand the long-term consequences. We are, in effect, making a permanent alteration to their genetic blueprint without their agreement. This raises fundamental questions about autonomy and our right to make choices for beings who cannot make choices themselves. Beyond the immediate lack of consent, there's the issue of unintended consequences. Our understanding of genetics is still evolving. Even with the most sophisticated tools, we can't predict with absolute certainty how a genetic change will interact with other genes, how it might affect development, or how it could ultimately manifest in future generations. A seemingly beneficial alteration could trigger unforeseen problems down the line, potentially creating new diseases or vulnerabilities. Furthermore, the complexity of the human genome means that modifying one gene could have cascading effects on numerous others, a butterfly effect on a scale we scarcely grasp. The concept of designer babies also arises, a term often associated with germline gene therapy, although it's important to approach it with caution. While the intention might be purely therapeutic, the possibility of using germline gene therapy for non-medical enhancements, altering traits like intelligence, athletic ability, or even appearance, is a deeply troubling one. This could exacerbate existing social inequalities, creating a genetically stratified society where advantage is determined at birth. It's a slippery slope, and careful consideration is needed to prevent such outcomes. Another significant consideration is the potential for germline modifications to be lost over time. Genes, like all biological information, can be subject to mutations, recombination, and other processes that can disrupt the intended change. If a correction is made, it's possible that, over many generations, it could be lost, requiring repeated interventions. This raises questions about the sustainability of germline gene therapy and the burden it could place on future generations. The philosophical implications are equally profound. Some argue that altering the germline represents a fundamental shift in our relationship with nature and with our own evolution. For millennia, humans have adapted to their environment through natural selection, a process of trial and error that has shaped our species. Germline gene therapy represents a deliberate, artificial intervention in that process, raising questions about whether we have the right to fundamentally alter the course of human evolution. International regulations surrounding germline gene therapy are currently very limited and vary considerably between countries. Some nations have explicitly banned any form of germline modification, while others have no specific laws in place. This lack of global consensus creates a significant challenge, as research in one country could easily be replicated in another. There's a pressing need for a robust, internationally agreed-upon framework that balances the potential benefits of germline gene therapy with the serious ethical risks. The discussion inevitably brings to mind the concept of heritable genetic variation, the natural diversity within a population. This variation is crucial for the survival and adaptation of our species. While eliminating inherited diseases is a noble goal, eliminating all genetic variation could make the human population more vulnerable to future environmental changes or novel diseases. Maintaining a degree of natural diversity is often seen as vital for resilience. Ultimately, the debate surrounding germline gene therapy is not simply a scientific one. It's a deeply human one, touching on fundamental questions about our values, our responsibilities, and our place in the world. It's a conversation that requires input from scientists, ethicists, policymakers, and the public. A conversation that must proceed with caution, humility, and a profound respect for the delicate balance of life itself. The promise of eradicating inherited diseases is powerful, but the potential consequences demand that we proceed with the utmost care, always remembering that the future we are shaping may belong not just to us, but to countless generations to come. The world of gene therapy has been quietly transforming, building upon decades of research into how to precisely introduce genetic instructions into cells. While earlier approaches focused on delivering intact DNA, a revolutionary method has emerged, particularly vital in the rapid response to global health challenges. This new strategy centers on messenger RNA, or mRNA, and its clever delivery using tiny, protective bubbles called lipid nanoparticles. These LNPs are the key to unlocking mRNA's therapeutic potential, and they've become absolutely critical in the development of the COVID-19 vaccines. mRNA itself isn't a gene, it's a blueprint, a set of instructions detailing how to build a specific protein. Remember those synthetic gene circuits we discussed? mRNA is similar. It carries the code for a protein, but unlike DNA, it doesn't integrate into your cell's existing genetic material. Instead, it acts as a temporary message, telling the cell's machinery to start building that protein. The protein, in turn, can then trigger an immune response, like fighting off a virus, or deliver a corrective signal within the body. The problem, however, was getting the mRNA safely into the cells in the first place. mRNA is fragile. It quickly breaks down in the body's environment. It needs a shield, a way to protect it from enzymes that would dismantle it before it could do its job. That's where lipid nanoparticles come in. These are the giant bubbles you might picture. They're incredibly small, only a few nanometers in diameter. A nanometer is 1/1000th of a meter. Think of them as miniature, highly sophisticated capsules. They're made of lipids, which are fats specifically, a blend of different types of lipids, including cholesterol, polar lipids like dysterophosphatidylcholine, DSPC, and sometimes polyethylene glycol, PEG. This carefully chosen combination creates a shell that's both strong and flexible, allowing the nanoparticle to navigate its way through the body's defenses. The lipid composition is incredibly important. The outer layer of the nanoparticle is designed to interact with the cell membrane, facilitating entry. The inner core of the nanoparticle holds the mRNA, carefully encapsulated to prevent degradation. Once the LNP reaches the target cell, say, a muscle cell or an immune cell, the outer lipid layer merges with the cell membrane, effectively becoming part of the cell's surface. This merging isn't a forceful collision. It's a gentle exchange, driven by the natural tendency of lipids to interact with each other. The nanoparticle essentially dissolves into the cell membrane, releasing the mRNA into the cell's cytoplasm, the fluid-filled space within the cell where many of its processes occur. Now let's talk about how this works specifically in the context of the mRNA vaccines. For the COVID-19 vaccines, the mRNA contained a blueprint for the spike protein found on the surface of the SARS-CoV-2 virus. When injected, the LNPs delivered this mRNA into cells, primarily immune cells like dendritic cells, prompting them to start producing the spike protein. This spike protein isn't harmful, it's just a signal. The immune system then recognizes this foreign protein as something it shouldn't be there. This triggers a cascade of events. The immune system produces antibodies, which are proteins that specifically target the virus, and it generates T cells, which can directly attack infected cells. This rapid, targeted immune response is what protects you from the virus. The beauty of using LNPs for mRNA delivery is that it's remarkably adaptable. The lipid composition can be tweaked to optimize the delivery for different cell types and different diseases. Researchers are exploring LNPs for gene therapy applications beyond vaccines, including treatments for cancer, genetic disorders, and even neurological diseases. For instance, LNPs can be engineered to target specific tissues, like the liver or the brain, ensuring that the therapeutic mRNA reaches the right place in the body. Furthermore, the LNP technology is incredibly scalable. The process of manufacturing LNPs is relatively straightforward, allowing for mass production, a critical factor, especially during a global pandemic. The speed with which LNPs could be produced and deployed was a key element in the rapid development and rollout of the COVID-19 vaccines. The ongoing research into LNPs also focuses on improving their stability and reducing potential side effects. Some early vaccine formulations raised concerns about inflammation, and researchers are actively investigating ways to minimize these effects. The composition of the LNPs, the amount of mRNA they carry, and the route of administration are all being carefully examined to optimize both efficacy and safety. Looking ahead, the future of gene therapy is undoubtedly intertwined with the continued advancements in LNP technology. As researchers gain a deeper understanding of how these nanoparticles interact with cells and the immune system, we can anticipate even more sophisticated and targeted therapies. The ability to deliver genetic instructions with such precision represents a monumental step forward, holding immense promise for treating a wide range of diseases and fundamentally changing how we approach healthcare. The journey of mRNA and LNPs is still unfolding, but it's a story filled with scientific innovation and the potential to revolutionize medicine. The battle against cancer has always been a complex one, a constant race between rapidly dividing cells and the body's defenses. For decades, treatments like chemotherapy and radiation have aimed to slow this growth, but they often attack healthy cells too, causing debilitating side effects. Scientists have been tirelessly exploring new approaches, and one particularly exciting area involves a clever strategy using viruses, but not the kind that make us sick. We're talking about oncolytic viruses, carefully engineered viruses designed to specifically target and destroy cancer cells while leaving healthy cells untouched. These aren't your average viruses. They're meticulously crafted, often derived from naturally occurring viruses, but with specific changes introduced to make them behave in a very particular way. The first step is usually to remove the genes that allow the virus to replicate and spread widely throughout the body. This is absolutely crucial. We don't want a normal viral infection. Instead, we're left with a virus that can infect cancer cells but doesn't create a widespread systemic infection. It's like giving a targeted weapon, not a cannon blast. Once the replication genes are removed, the virus's main function, to infect and eventually destroy the cancer cell, remains. This infection process itself is incredibly powerful. When an oncolytic virus reaches a cancer cell, it uses specific receptors like little docking stations that are often overexpressed on the surface of those cancerous cells. These receptors are like hello signals, and the virus is programmed to recognize and bind to them. It's a remarkably precise way of locating its target. Once attached, the oncolytic virus gains entry into the cancer cell. Inside, the virus unleashes a cascade of events designed to destroy the cell from within. It can directly disrupt cellular processes like DNA replication, preventing the cancer cell from dividing and multiplying. More impressively, the infection triggers the body's own immune system. As the virus replicates inside the cancer cell, it releases fragments of itself, called viral antigens. These antigens are recognized as foreign by the immune system, stimulating a powerful immune response. This response involves activating T cells, specialized immune cells that are experts at hunting down and eliminating infected cells. Essentially, the virus isn't just killing the cancer cell directly. It's training the immune system to recognize and attack cancer cells in the future. Different types of oncolytic viruses are being explored. Adenoviruses, which are commonly used in some vaccines, have been modified to specifically target tumors. Herpes simplex virus type 1, HSV1, known for causing cold sores, is another candidate, and researchers are also investigating modified poliovirus and vaccinia virus, the virus used in the original smallpox vaccine. Each virus has its own unique advantages and disadvantages, and scientists are constantly refining their designs to improve their efficacy and safety. The beauty of this approach isn't just in the virus itself, but also in the potential for combination therapies. Oncolytic viruses can be used alongside other cancer treatments, such as chemotherapy or radiation therapy, to enhance their effectiveness. They can also be combined with immune checkpoint inhibitors, drugs that block the mechanisms cancer cells use to evade the immune system. This coordinated attack significantly boosts the chances of success. Researchers are also working on ways to deliver these oncolytic viruses directly to the tumor site. This could involve injecting them directly into the tumor, or using techniques like focused ultrasound to temporarily open blood vessels and allow the viruses to reach the tumor more effectively. Another strategy involves engineering the viruses to produce therapeutic molecules within the tumor, further amplifying their destructive power. Clinical trials are underway, assessing the safety and effectiveness of various oncolytic virus therapies for different types of cancer, including melanoma, head and neck cancer, and pancreatic cancer. Early results are promising, demonstrating the ability to shrink tumors and, crucially, to elicit a strong anti-tumor immune response. It's important to remember that these are still early stages, and a significant amount of research is needed before these therapies become widely available. However, the potential of oncolytic viruses to revolutionize cancer treatment is truly remarkable. They represent a shift in thinking, moving beyond simply suppressing cancer growth to actively harnessing the body's own defenses. The continued development of these therapies hinges on understanding the complex interactions between the virus, the cancer cell, and the immune system. Scientists are meticulously studying these interactions to identify new targets and refine the virus designs for maximum impact. The ultimate goal is to create highly specific, potent oncolytic viruses that can selectively destroy cancer cells while minimizing harm to healthy tissues, offering a genuinely personalized and powerful weapon in the fight against this devastating disease. It's a field brimming with innovation and hope, driven by the unwavering determination to conquer cancer, one carefully engineered virus at a time. The world of bleeding isn't a silent one. It's a world of tiny, insistent drips, a constant, frustrating reminder of a body's inability to do what it should, to stop the flow of blood when a vessel is damaged. For individuals living with hemophilia, this is a reality far beyond a minor inconvenience. It's a condition rooted in a fundamental problem, a lack of specific clotting factors, the proteins that trigger a cascade of events leading to the formation of a stable blood clot. Hemophilia, particularly the most common type, hemophilia A, arises from mutations in the gene responsible for producing factor VIII, one of the key players in this intricate process. This missing or malfunctioning factor disrupts the entire clotting system, making even minor injuries profoundly problematic. For decades, the treatment of hemophilia relied heavily on frequent infusions of factor VIII concentrates. These concentrates, derived from human blood donations, provided temporary relief, mimicking the body's own ability to clot. However, this approach was far from ideal. The concentrates were expensive, carried the risk of allergic reactions, and could sometimes trigger immune responses that ultimately led to the body developing antibodies against factor VIII, diminishing the effectiveness of future treatments. Scientists recognized that a more durable and precise solution was needed, and that solution began to take shape with the burgeoning field of gene therapy. The core concept behind gene therapy for hemophilia is surprisingly elegant. Instead of simply supplying a missing protein, it aims to correct the underlying genetic defect. Researchers began to explore methods of delivering a healthy copy of the factor VIII gene directly into the patient's cells, specifically the liver cells, which are the primary site of factor VIII production. The liver, you see, is naturally designed to manufacture clotting factors, but in individuals with hemophilia A, this production is hampered by the defective gene. Several approaches have been developed, each with its own nuances and challenges. One of the earliest and most successful methods involves the use of an adeno-associated virus, AAV, as a vector, a tiny, harmless virus engineered to carry the therapeutic gene. AAVs have a unique advantage. They are naturally well tolerated by the human body and don't trigger strong immune responses. The AAV is carefully modified to remove any harmful genes and is loaded with a functional copy of the factor VIII gene. Once delivered into the liver cells, this new gene begins to instruct the cells to produce factor VIII. It's a slow process, taking time for the cells to transcribe the gene into messenger RNA, which then directs the creation of the clotting factor protein. The liver cells then start releasing the factor VIII into the bloodstream, effectively restoring the patient's clotting ability. The beauty of this approach is its potential for long-term efficacy. Unlike temporary infusions, the body now produces its own factor VIII, significantly reducing the need for external treatments. While initial results were promising, achieving consistent and sustained factor VIII levels in the bloodstream presented a hurdle. Researchers diligently worked to refine the delivery methods and optimize the expression of the therapeutic gene. Significant advancements were made by tweaking the AAV vector itself. Different serotypes of AAVs were tested, each with varying abilities to infect liver cells and to maintain the long-term expression of the factor VIII gene. Researchers also investigated ways to boost the production of factor VIII within the cells. One particularly interesting strategy involved using small molecules, carefully selected chemicals, to enhance the activity of the gene expression machinery within the liver cells. These molecules essentially wake up the gene, encouraging it to produce more factor VIII. The success of gene therapy for hemophilia has been demonstrated in clinical trials across the globe. Patients who received AAV-mediated gene therapy have shown remarkable improvements in their clotting ability, with many achieving sustained factor VIII levels that allow them to lead relatively normal lives. Some patients have been able to stop taking regular factor VIII infusions entirely, a truly transformative outcome. It's crucial to understand that gene therapy isn't a one-and-done solution. Monitoring the patient's factor VIII levels remains essential, and occasional supplemental treatments may still be needed, particularly during periods of stress or injury. However, the frequency and dosage of these treatments are significantly reduced compared to traditional factor VIII therapy. Moreover, research continues to expand the possibilities. Scientists are exploring ways to not only deliver the factor VIII gene, but also to enhance its expression and durability within the liver. This includes investigating alternative gene delivery methods and developing strategies to overcome potential immune responses. The journey of gene therapy for hemophilia has been a testament to human ingenuity and the power of scientific collaboration. It represents a shift from managing the symptoms of the disease to addressing its root cause, offering hope and a brighter future for individuals affected by this challenging condition. The ongoing advancements in this field are steadily paving the way for even more effective and personalized treatments, solidifying gene therapy's position as a truly groundbreaking approach to health care. The initial excitement surrounding early gene therapy trials often centered on the immediate, observable effect: a sudden improvement in a patient's condition, a temporary reduction in symptoms. These were undeniably encouraging signs, demonstrating the potential of this revolutionary approach. However, the real story of gene therapy's success, and indeed its future, lies far deeper, woven into the intricate fabric of long-term observation and the establishment of truly durable expression of the therapeutic gene within the patient's body. It's like planting a beautiful tree. The initial sprouting and growth are wonderful, but the tree's health and longevity depend on consistent care, proper nourishment, and protection from the elements. Gene therapy is no different. The challenge with many early gene therapies stemmed from the transient nature of the introduced genetic material. Once the vector, the delivery system, be it a viral particle or a lipid nanoparticle, had done its job and deposited the gene into the cell, the cell's natural processes often led to degradation or silencing of the new genetic code. The cell, recognizing a foreign element, would activate defense mechanisms designed to eliminate it, preventing any lasting change. This meant that while a patient might experience a positive response after a single treatment, the effect was often short-lived, requiring repeated administrations to maintain the benefit. This wasn't a failure of the gene therapy itself, but rather an indication that the delivery system hadn't provided a sustainable solution. Researchers quickly realized that simply delivering a gene wasn't enough. They needed to engineer the process to ensure its sustained presence and functionality within the target cell. This is where the concept of durable expression became paramount. Several strategies were developed to achieve this. One key technique involved selecting and modifying vectors to enhance their integration into the host cell's genome. Some viral vectors, for example, are engineered to integrate themselves randomly into the chromosome, often near genes that are already active and therefore supportive of the new gene's expression. This meant the cell didn't view the introduced gene as a foreign invader, but rather as a natural component of its own genetic landscape. Furthermore, scientists began focusing on the copy number of the therapeutic gene. A single copy of the gene might quickly be silenced, but introducing multiple copies, either through the vector design or through techniques like using a self-replicating gene, dramatically increased the chances of sustained expression. This is analogous to having several backup hard drives in a computer. If one fails, the other remains operational. The more copies of the therapeutic gene, the greater the capacity for the cell to produce the desired protein. However, simply increasing the number of copies wasn't without its complexities. In some cases, a large number of copies could trigger unintended consequences, such as disrupting the normal regulation of other genes. Therefore, careful design and meticulous evaluation were crucial. Sophisticated monitoring techniques were developed to track the levels of the therapeutic protein being produced and to identify any potential off-target effects. Long-term monitoring became, and continues to be, a cornerstone of gene therapy research. It's not enough to simply observe the immediate response. Researchers now routinely follow patients for years, sometimes decades, collecting detailed data on the protein levels, the patient's overall health, and any potential adverse effects. This longitudinal data is absolutely critical for understanding the true durability of the treatment. Researchers often use sophisticated imaging techniques, like PET scans, to visualize the distribution of the therapeutic gene within the body and to assess its impact on various tissues. Another vital aspect of durable expression revolves around the concept of epigenetic modifications. Epigenetics refers to changes in gene expression that don't involve alterations to the underlying DNA sequence itself. These changes, such as DNA methylation or histone modification, can influence whether a gene is switched on or off. Early gene therapies often overlook the impact of these modifications. Now, research is exploring ways to engineer vectors that can subtly modulate the epigenetic landscape, effectively teaching the cell to consistently express the therapeutic gene. This involves utilizing molecules that influence the epigenetic machinery, creating a more stable and predictable environment for the gene's continued activity. The data emerging from long-term monitoring studies is painting an increasingly optimistic picture. In some trials, patients receiving gene therapies for conditions like spinal muscular atrophy have demonstrated sustained improvement for years, with protein levels remaining remarkably stable. While challenges remain, particularly with complex diseases and tissues, the ability to track outcomes over extended periods is transforming our understanding of gene therapy's potential. Moreover, advances in vector design and manufacturing are continually improving the precision and stability of gene delivery. Newer vectors are engineered to minimize immune responses, increasing the chance of sustained expression by preventing the body from attacking and eliminating the vector and its payload. The development of split delivery strategies, where multiple vectors are used to target different cell types, is also proving effective in achieving more comprehensive and durable expression. Ultimately, the journey of gene therapy is one of continuous refinement, driven by meticulous observation and a deep understanding of cellular biology. It's not a set-it-and-forget-it process. Rather, it's an ongoing dialogue between the therapeutic gene and the host cell, constantly adapting and evolving towards a state of harmonious coexistence. The future of gene therapy hinges on our ability to not only deliver genes effectively, but to ensure their long-term, reliable expression, allowing us to truly unlock the transformative potential of this powerful technology. The world of genetic diseases is a complex one, a universe of tiny mistakes within our instructions for building our cells. Some of these errors are easily corrected, like a simple typo in a book, but others are incredibly stubborn, deeply woven into the very fabric of our DNA, leading to conditions that can cause immense suffering. One of the most heartbreaking of these is Duchenne muscular dystrophy, or DMD. It's a disease where the muscle fibers simply stop working, slowly but surely, robbing people of their movement and strength. DMD is caused by a mutation, a change, in a specific part of the gene called dystrophin. Dystrophin is incredibly important. It's like a crucial support system for muscle cells, holding them together and allowing them to contract. Without it, muscles become weak and eventually can't function. For decades, researchers have been trying to find ways to fix this genetic problem. Early approaches focused on replacing the entire dystrophin gene, a monumental task given its enormous size. However, delivering such a large piece of DNA into muscle cells proved incredibly difficult, often resulting in only short-term expression, if any at all. More recently, scientists have shifted their attention to a smaller, less obvious area within the dystrophin gene, the untranslated regions, or UTRs. These regions are found at the beginning and end of the gene, and unlike the coding sequence itself, they don't contain instructions for building the protein. Instead, they play a surprisingly significant role in controlling how the gene is read and used. The UTRs are like little messengers, directing the machinery that reads the gene's instructions. They help to ensure that the correct amount of dystrophin is made, and they also protect the gene from being damaged. In DMD, disruptions within these UTRs can lead to reduced dystrophin production, further worsening the disease. Researchers have begun exploring ways to target these UTRs to restore dystrophin expression. One strategy involves modifying the 5-UTR, the region at the beginning of the gene. Scientists have discovered that certain sequences within this UTR can hinder the ability of the cell to properly read the gene. By introducing short sequences into the UTR that counteract this effect, they've been able to improve dystrophin production in laboratory studies. Think of it like adding a clear instruction manual to the gene, telling the cell exactly how to build the protein correctly. Another area of intense investigation is the 3-UTR, the region at the end of the gene. This UTR is particularly vulnerable to mutations in DMD. It plays a key role in exporting the newly made dystrophin protein out of the cell, and when this process is disrupted, the protein builds up inside the cell and eventually gets degraded. Researchers are designing molecules that can specifically target and correct these problems. These molecules, often called antisense oligonucleotides, are short strands of DNA or RNA that bind to the faulty sequences in the UTR, preventing them from causing harm. A particularly exciting approach involves using CRISPR-Cas9 technology, a revolutionary gene editing tool. While CRISPR is often associated with directly changing the DNA sequence itself, it can also be used to modify the UTRs. Using CRISPR, scientists can precisely target and alter the sequences within the UTR, effectively rewriting the instructions and improving dystrophin production. This method offers incredible precision and the potential to address multiple mutations simultaneously, which is a significant advantage. The beauty of targeting the UTRs lies in several factors. The UTRs are significantly smaller than the dystrophin gene itself, making delivery much easier. Viral vectors, the delivery vehicles used in many gene therapies, can carry these UTR-modifying molecules efficiently. Moreover, the UTRs are relatively stable, meaning that the therapeutic effects can potentially last longer than those achieved by directly modifying the coding sequence. Preclinical studies, experiments done in the lab with cells and animals, have shown remarkable results. Mice with DMD symptoms have experienced improved muscle strength and function after receiving UTR-targeting gene therapy. These improvements weren't just temporary. In some cases, the effects persisted for months, a significant advancement compared to earlier gene therapy attempts. However, significant challenges remain. Ensuring that the UTR-modifying molecules reach the correct muscle cells is crucial. The body's immune system can also recognize these foreign molecules as invaders, triggering an immune response that can reduce their effectiveness. Scientists are developing strategies to minimize this immune response, such as using immune-modulating drugs or engineering the UTR-modifying molecules to be less recognizable by the immune system. Despite these hurdles, the focus on the UTRs represents a potentially transformative shift in the treatment of DMD. It's a testament to the ingenuity of scientists and their persistent efforts to unravel the complexities of genetic diseases. The journey to delivering effective UTR-targeted gene therapy is far from over, but the early results offer a glimmer of hope for individuals and families affected by this devastating condition, offering a more precise and potentially longer-lasting solution. It's a complex story, but one filled with possibility. The world of genetic disease is a remarkably intricate one, filled with tiny mistakes in our instructions for building and operating our bodies. Some of these mistakes cause serious problems, affecting everything from how our blood clots to how we move. Today, we're going to explore a particularly promising area of gene therapy focused on a condition called sickle cell disease. This disease isn't about a single broken gene, but rather a subtle shift in how a key part of our red blood cells functions. Sickle cell disease arises from a mutation, a change, within the beta-globin gene. This gene is responsible for producing a protein called beta-globin, which is a crucial component of hemoglobin. Hemoglobin, you'll remember, is the protein found inside our red blood cells that carries oxygen throughout our bodies. The normal beta-globin gene contains a specific sequence of DNA letters. In individuals without sickle cell disease, this sequence dictates the production of a perfectly functioning beta-globin protein. However, in people with sickle cell disease, a single DNA letter, the sixth codon in the beta-globin gene, has been changed. Instead of the normal DNA code for glutamic acid, this mutated gene produces a different amino acid, valine. This seemingly small alteration has a huge impact. The resulting beta-globin protein is less stable, and when combined with other factors, causes the hemoglobin molecules to clump together, distorting the shape of the red blood cells into a crescent or sickle shape. These sickle-shaped cells are fragile and have difficulty navigating the tiny capillaries in our tissues, leading to pain, organ damage, and a shortened lifespan for the affected red blood cells. The constant destruction of these cells forces the body to work overtime, producing immature red blood cells and ultimately causing anemia. The clinical manifestations of sickle cell disease can vary greatly, with some individuals experiencing severe symptoms from infancy, while others may have milder symptoms that develop later in life. Now, scientists are exploring gene editing techniques to directly correct this single mutation. Several approaches are being investigated, but the most prominent utilizes CRISPR-Cas9 technology. CRISPR-Cas9 functions like a pair of molecular scissors, guided by a small piece of RNA that can precisely locate and cut DNA at a specific location. In the context of sickle cell disease, the CRISPR-Cas9 system is designed to target the mutated beta-globin gene and replace it with the correct, functional sequence. The process begins with a healthy copy of the beta-globin gene, also packaged within a tiny, harmless virus, which acts as a delivery vehicle. This virus is carefully engineered to only infect the patient's own red bone marrow cells, the birthplace of all blood cells. Once inside these cells, the CRISPR-Cas9 system, guided by the RNA, finds the mutated gene and makes the precise cut. This cut initiates the cell's natural DNA repair mechanisms. Because the cell's own repair machinery is activated, it naturally replaces the mutated sequence with the correct beta-globin gene. Importantly, this isn't simply inserting a new gene, it's correcting the existing one, offering a more stable and reliable outcome. The healthy beta-globin protein then begins to be produced, restoring the normal function of the red blood cells. Researchers are also exploring different methods of delivering the CRISPR-Cas9 system. One approach involves directly injecting the components of the system, the Cas9 enzyme and the guide RNA, directly into the bone marrow. Another method utilizes modified viruses that can efficiently deliver the CRISPR-Cas9 machinery to the target cells. The choice of delivery method is a critical factor, impacting the efficiency and safety of the gene editing process. The beauty of this approach lies in its potential for a long-term solution. Unlike traditional gene therapy, which often relies on introducing a new gene gene editing seeks to fix the existing genetic defect. This dramatically reduces the risk of the body mounting an immune response against the corrected gene, a common challenge in gene therapy. Furthermore, researchers are diligently studying the long-term effects of this gene editing treatment. Careful monitoring is paramount to ensure the corrected gene remains active and doesn't inadvertently cause any unwanted changes. Scientists are tracking the production of normal hemoglobin, the health of the modified red blood cells, and the overall response of the patient's immune system. Early clinical trials of this gene editing approach have yielded encouraging results. Patients who undergo this treatment experience a significant reduction in the number of sickle cells in their blood, and many report a dramatic decrease in pain crises, the excruciating episodes of pain that characterize this disease. Importantly, the edited cells persist within the bone marrow for many years, providing sustained correction of the genetic defect. The journey towards a curative treatment for sickle cell disease is still ongoing, but the potential of gene editing represents a monumental step forward. It's a testament to the power of scientific innovation and a beacon of hope for millions of individuals and families affected by this debilitating disease. It offers not just symptom relief, but the possibility of a truly healthy and normal life, built on a foundation of correctly functioning red blood cells. The precision and durability of this approach are redefining the possibilities within the field of gene therapy, and its continued development holds immense promise for countless others facing genetic challenges. The creation of a gene therapy product isn't simply about delivering a gene into a patient's cells. It's a remarkably intricate process, much like building a very precise, incredibly small and incredibly sensitive machine. At its heart, the gene itself needs a vehicle, a vector, to safely and effectively reach the target cells. These vectors, most commonly derived from modified viruses, are carefully engineered to carry the therapeutic gene and then disappear once their job is done. The challenge then becomes how to produce these vectors, initially in small quantities, and then, crucially, to scale up the production to meet the needs of clinical trials and, eventually, widespread use. Early production of viral vectors like adenoviruses or adeno-associated viruses, AAVs, relied heavily on cell culture. Specialized cell lines, often human or simian cell lines, were grown in large flasks or bioreactors. These cells were meticulously controlled: temperature, pH, oxygen levels. Everything had to be perfect to support the efficient replication of the viral vector. The virus then naturally assembled itself within these cells and was separated from the cell through a series of careful filtration and purification steps. Think of it like brewing a very complicated tea, where every ingredient and every step needs to be absolutely precise to achieve the desired flavor. However, scaling up this process presented immediate hurdles. The initial small-scale production, while necessary for preclinical testing and early clinical trials, simply couldn't produce enough virus to treat even a small group of patients. The growth rates of the cells themselves were also a limiting factor. Scaling up cell culture requires enormous bioreactors, huge stainless steel tanks, sometimes the size of small rooms, where the cells are grown in massive quantities. Maintaining sterility in these large vessels is incredibly difficult, demanding sophisticated filtration systems and rigorous cleaning protocols. Any contamination could ruin an entire batch of viral vectors, leading to significant financial losses and delaying patient treatment. The purification of the vectors was another significant bottleneck. Separating the desired viral vector from all the cellular debris and other proteins produced by the cells is a complex process involving multiple chromatography steps. Essentially, using different substances to selectively bind to the vector and wash away impurities. Each step adds to the time and cost, and optimizing these purification methods for larger-scale production was a considerable undertaking. Furthermore, the size of the vectors themselves, particularly AAVs, meant that even after purification, a significant amount of material could remain bound to chromatography columns, reducing the overall yield. The cost of producing viral vectors is, frankly, substantial. It's not just the cost of the raw materials, the cell culture media, the chromatography resins, the specialized equipment. There are also the significant costs associated with quality control, extensive testing, to ensure the vectors are safe, effective, and free from contamination. These tests include assessing the vector's titer, the number of viral particles per volume, and conducting sterility assays, endotoxin assays, and capsid analysis, examining the outer shell of the virus. Each of these tests requires specialized equipment and trained personnel, adding considerably to the overall expense. As gene therapy has moved closer to widespread clinical use, considerable investment has been directed towards developing more efficient and automated production methods. One promising approach involves using transient transfections, a technique where cells are temporarily charged with genetic material without permanently integrating it into their DNA. This avoids the complexities and potential risks associated with integrating vectors into the host genome. Another area of focus is continuous manufacturing, where the production process is continuously running, reducing downtime and increasing overall throughput. Process analytical technology, PAT, is also playing a growing role. PAT involves using sensors and data analysis to monitor the production process in real time, allowing for immediate adjustments to maintain optimal conditions and ensure consistent vector quality. This moves the manufacturing process away from being reactive, responding to problems after they arise, and towards being proactive, anticipating and preventing issues before they impact product quality. Furthermore, there's a shift towards using GMP-compliant, good manufacturing practice facilities, which adhere to strict regulations established by regulatory agencies like the FDA. These facilities are designed and operated to ensure the highest levels of quality control and traceability throughout the entire production process. The transition to a GMP-based manufacturing model is crucial for building confidence in the safety and efficacy of gene therapy products. The economics of gene therapy production are constantly evolving as technology advances. While the initial cost per dose was extraordinarily high, advancements in automation, process optimization, and manufacturing scale are steadily driving down the cost. The future of gene therapy production likely involves a combination of continuous manufacturing, automated quality control systems, and sophisticated data analytics to create a robust and cost-effective supply chain. It's a fascinating and complex field, a blend of biology, engineering, and meticulous manufacturing, all working together to bring these revolutionary treatments to patients who desperately need them. The world is a symphony of light, and for many, the ability to see it fully is a fundamental part of experiencing it. But for individuals living with inherited blindness, that symphony is muted, the notes of color lost to a genetic quirk. A significant portion of inherited blindness arises from defects affecting the retinal pigment cells, RPs, which reside within the retina, the light-sensitive tissue at the back of the eye. These cells, unlike the photoreceptors, rods, and cones that directly detect light, play a crucial supporting role, and scientists have been diligently exploring gene therapy as a potential pathway to restore their function. The retina relies on RPs to maintain a healthy environment. These cells produce melanin, the same pigment that gives our skin its color, which acts as a natural sunscreen, protecting the photoreceptors from damaging ultraviolet rays. They also produce vital growth factors and neurotransmitters, essentially communicating with the photoreceptors and ensuring the overall health and efficiency of the visual system. When a gene responsible for RP function is mutated, the cells struggle to perform these tasks properly, ultimately impacting the photoreceptor's ability to operate effectively. Several gene therapy approaches are being investigated specifically targeting RPs in conditions like retinitis pigmentosa, RP, a group of inherited diseases causing progressive vision loss. One prominent strategy focuses on delivering a functional copy of the mutated gene directly into the RPs. This is achieved using adeno-associated viruses, AAVs, tiny viruses engineered to be harmless and capable of carrying genetic material into cells. AAVs are particularly well suited for gene therapy because they efficiently infect cells and, importantly, don't typically trigger a strong immune response, a significant hurdle in gene therapy. Researchers carefully select AAV serotypes, different versions of the virus, that preferentially infect RPs, maximizing the chances of the therapeutic gene taking hold. Another exciting area of research involves introducing genes that boost the RP's own production of essential proteins. For instance, some forms of RP are linked to a deficiency in transthyretin TTR, a protein crucial for RP survival. Gene therapy with AAVs can deliver a functional TTR gene, allowing the RPs to manufacture sufficient amounts of this vital protein. This isn't just about providing a new gene. It's about reactivating the RP's inherent ability to self-repair and maintain their core functions. Beyond simply delivering a new gene, researchers are also exploring strategies to enhance the RP's existing machinery. Certain genetic mutations disrupt the RP's ability to process and respond to signals from the photoreceptors. Gene therapy can introduce genes that correct these processing errors, improving the communication pathways within the retina. This represents a more nuanced approach, tackling the root cause of the dysfunction rather than simply replacing a missing component. Furthermore, scientists are investigating the possibility of switching on dormant genes within the RPs. Some RPs are linked to genes that have been silenced due to genetic changes. Gene therapy could be used to reactivate these silenced genes, restoring their function and potentially reversing the symptoms of the disease. This concept, known as gene reactivation, is a particularly promising avenue of research. The delivery method itself is continually being refined. Current techniques often involve a subretinal injection, where the AAV vector is directly injected into the space beneath the retina. Scientists are working to improve the precision and safety of this injection process, minimizing potential side effects such as inflammation or immune responses. They're also exploring alternative delivery methods, like injecting the vector into the bloodstream and allowing it to cross the blood-retina barrier, a tightly controlled membrane that protects the eye from harmful substances. Preclinical studies involving cell cultures and animal models are crucial steps in evaluating the safety and efficacy of these gene therapy approaches. These studies help researchers understand how the AAV vectors interact with the RPs, identify potential adverse effects, and determine the optimal dosage. Detailed imaging techniques, such as optical coherence tomography, OCT, allow scientists to visualize the distribution of the AAV vectors within the retina and assess the impact on RP function. Clinical trials, when deemed safe and promising, represent the next stage in this journey. These trials are carefully designed to evaluate the long-term safety and effectiveness of the gene therapy approach in human patients. While challenges remain, including the complexity of the eye and the potential for immune responses, the progress made in gene therapy for inherited blindness, particularly targeting RPs, is incredibly encouraging. The ability to harness the power of genetic manipulation to restore function to these vital cells offers a glimmer of hope for individuals affected by these devastating conditions. The journey is complex, requiring meticulous research and careful execution, but the potential to unveil the full beauty of the visual world for those who have lost it is a truly remarkable goal. The human body is a marvelous, intricate machine, and sometimes tiny errors can creep into the instructions that guide its operation. These errors can manifest as diseases, disorders that disrupt the normal processes within our cells. While gene editing, as we've explored, focuses on directly correcting these faulty genetic blueprints, another incredibly clever strategy is emerging, a technique called RNA interference, or R9. It's a subtle but powerful way to influence how our genes behave, without fundamentally changing the DNA itself. R9 works by harnessing the cell's own natural defense mechanisms. Every gene contains instructions for building proteins. These instructions are written in a language called messenger RNA, or mRNA. mRNA carries the genetic code from the DNA to the protein-making machinery of the cell. However, mRNA isn't permanent. The cell quickly breaks down mRNA once its instructions have been used. Now, scientists discovered that tiny strands of RNA, specifically small interfering RNAs, or cRNAs, could recognize and bind to specific mRNA molecules. Think of cRNAs as tiny, highly precise detectives. When a cRNA finds its matching mRNA target, it triggers a process that ultimately leads to the destruction of that mRNA. Because the mRNA is gone, the protein it would have produced is never created. This is essentially turning off the gene's activity. The beauty of RNAi lies in its specificity. Scientists can design cRNAs to target nearly any gene they want to silence. It's like creating a custom off switch for a particular protein. The cRNA is carefully crafted to bind only to the mRNA of the gene causing the problem, minimizing the risk of unintended consequences. This remarkable accuracy is what makes RNAi such a promising tool in gene therapy. Let's consider a specific example: Huntington's disease. This devastating neurodegenerative disorder is caused by an abnormally long version of a gene that produces a protein called huntingtin. This faulty huntingtin protein accumulates in brain cells, leading to their death. While gene editing might be considered to correct the DNA sequence, RNAi offers a different approach. Scientists have developed cRNAs that specifically target the mRNA produced by the huntingtin gene, preventing the production of the toxic protein. Similarly, RNAi is being explored as a treatment for certain cancers. Many cancers are driven by genes that promote uncontrolled cell growth. By using cRNAs to silence those genes, researchers are trying to slow down or even halt the growth of cancerous tumors. It's important to note that this isn't about permanently eliminating the gene. It's about temporarily dampening its activity, giving the body's immune system a better chance to fight the disease. The development of RNAi therapeutics isn't without its challenges. One of the biggest hurdles is delivery. Getting the cRNAs to the right cells within the body is crucial. Just like delivering a package to the correct address, we need a reliable system to transport the cRNAs to the cells where they're needed. Researchers are exploring various delivery methods, including viral vectors, similar to those used in traditional gene therapy, and lipid nanoparticles, tiny bubbles that can encapsulate and deliver the cRNAs. Another concern is the duration of the effect. cRNAs are naturally broken down by the cell. So, researchers are working on strategies to prolong their activity, such as using modified cRNAs that are more resistant to degradation, or by developing systems that continuously deliver the cRNAs to the target cells. Beyond Huntington's disease and cancer, RNAi is showing potential in treating a wide range of genetic disorders, including cystic fibrosis, muscular dystrophy, and even certain viral infections. The ability to silence genes offers a truly personalized approach to medicine, tailoring treatments to the specific genetic makeup of each patient. It's a shift from simply correcting errors in DNA to gently modulating the activity of genes once they've already been expressed. The field of RNAi therapy is still relatively young, but the progress so far is incredibly exciting. Scientists are continually refining the technology, improving delivery methods, and expanding the range of diseases that can be targeted. It represents a fundamentally new way of thinking about gene therapy, one that leverages the cell's own sophisticated defense mechanisms to achieve therapeutic benefit. The intricate dance between RNA and DNA, once just a theoretical concept, is now transforming the landscape of medicine, offering hope for patients facing previously untreatable genetic conditions. The initial wave of gene therapy trials focused on delivering a single, corrected gene to as many patients with a particular disease as possible. It was like sending out a general message, hoping that enough people would receive the right instruction to fix their problem. This approach, while providing hope and demonstrating the potential of the technology, quickly revealed a crucial limitation. Genetic diseases aren't always identical. Even within the same disease, like cystic fibrosis or sickle cell anemia, the underlying genetic variations could be incredibly diverse. A single gene replacement might work beautifully for one patient, offering a complete, healthy version of the gene, but be completely ineffective for another. This wasn't a simple matter of dosage. The differences were encoded within the genes themselves. The realization dawned that a truly effective treatment wouldn't be a one-size-fits-all solution. Instead, the future of gene therapy lay in personalization, tailoring the treatment to an individual's unique genetic fingerprint. This shift marked the beginning of what's now known as personalized gene therapy, or sometimes precision gene therapy. The first step involved meticulously mapping the specific mutations responsible for a patient's disease. This went far beyond simply identifying the gene in question. It meant pinpointing the exact change within the gene sequence that was causing the problem. For example, in cystic fibrosis, numerous mutations in the CFTR gene can cause varying degrees of disease severity. Identifying the specific mutation, like the F508 deletion, the most common, became paramount. The technology needed to do this was rapidly evolving. Advanced DNA sequencing techniques, like whole exome sequencing and whole genome sequencing, became increasingly affordable and powerful. These methods could read the entire genetic code of a patient's cells, revealing not just the disease-causing mutation, but also other genetic variations that might influence how the body responds to the treatment. Some of these background genes, often called modifier genes, could amplify or diminish the effect of the corrected gene. Imagine a tuning fork. The corrected gene is the fundamental note, but the surrounding variations are the subtle adjustments that determine the tone and clarity. Researchers began to explore different ways to design gene therapies based on this individualized information. One approach involved designing gene therapy vectors, the vehicles that deliver the corrected gene, to recognize and bind specifically to the patient's mutated gene. Instead of simply inserting the healthy gene into the genome, the vector would be engineered to target only the damaged copy. This significantly reduced the risk of unintended consequences, such as disrupting other genes or triggering an immune response. For sickle cell anemia, for instance, Researchers developed vectors that specifically targeted the mutated beta-globin gene, promoting the production of the normal hemoglobin protein. Another critical development was the concept of gene editing, using tools like CRISPR-Cas9. While CRISPR offered incredible precision in correcting the DNA sequence directly, it also opened up possibilities for personalized gene editing. Rather than simply delivering a new gene, the system could be programmed to cut out the mutated section and replace it with the correct sequence. The beauty of this was that the editing process could be adapted to the unique mutation present in each patient's genome. This was particularly significant for diseases caused by small point mutations, changes in a single DNA building block, which are incredibly common. Furthermore, researchers started investigating ways to use a patient's own cells as the factory for producing the therapeutic protein. This is known as ex vivo gene therapy. In this approach, a patient's cells are removed from their body, genetically modified in a laboratory, and then returned to the patient. For instance, in treating severe combined immunodeficiency, SCID, hematopoietic stem cells, the cells that give rise to all blood cells were taken from the patient, modified to produce a functional immune system, and then infused back into the patient. The customization here was fundamental. The modifications were tailored to the patient's specific genetic defect. The development of personalized gene therapy is a complex and ongoing process. It requires sophisticated genetic analysis, innovative vector design, and a deep understanding of how genes interact with each other. There are still significant challenges to overcome, including the delivery of gene therapy vectors to the correct cells, ensuring long-term gene expression, and managing potential immune responses. However, the progress made in recent years is truly remarkable. As technologies continue to advance and our understanding of the human genome deepens, personalized gene therapy is poised to revolutionize the treatment of genetic diseases. It's not just about replacing a faulty gene. It's about understanding the intricate complexities of each individual's genetic makeup and designing a treatment that is perfectly suited to their unique needs. This represents a shift from treating the symptoms of a disease to addressing the root cause, the specific genetic defect that is driving the illness. The future of gene therapy is undeniably personalized, offering the potential to cure diseases that were once considered incurable. The journey is far from over, but each step forward brings us closer to a world where genetic diseases are no longer a life sentence, but rather a challenge that can be overcome with the precision and power of personalized gene therapy. The field of gene therapy is constantly evolving, driven by a desire to deliver precise genetic changes with ever-increasing efficiency and safety. While current methods, utilizing viral vectors like adeno-associated viruses, AAVs, have shown remarkable promise, scientists are now focusing on building vastly improved delivery systems and exploring how to combine gene therapies with other treatments for truly transformative effects. A lot of the current excitement centers around the development of next-generation vector technologies, pushing the boundaries of what's possible in targeting and inserting genes into specific cells. One particularly promising area involves the utilization of engineered viral vectors, not just as they are today, but as sophisticated tools designed to navigate the complex landscape of the human body. Researchers are working on vectors that can recognize and bind to specific receptors on the surface of target cells with incredible accuracy. Think of it like a key fitting perfectly into a lock, except instead of a lock, it's a receptor, and the key is a customized viral vector designed to deliver a therapeutic gene only to the cells that need it. This precision minimizes off-target effects, reducing the risk of unintended consequences. Furthermore, scientists are exploring vectors derived from different viruses altogether, investigating the potential of lentiviruses and herpaviruses, each with unique characteristics that could be optimized for particular gene therapies. The aim is to move beyond the limitations of AAVs, particularly regarding their packaging capacity, the amount of genetic material they can carry, and their ability to efficiently integrate their genetic cargo into the host cell's genome. Beyond simply improving vectors, considerable research is dedicated to altering the vectors themselves after they've delivered their genetic payload. Switchable vectors are being developed that can be activated only after reaching the target tissue. This is incredibly important, especially for conditions where the gene needs to be expressed for a limited period. Imagine a vector programmed to release its therapeutic gene only when it encounters a specific protein unique to a diseased cell, like a marker that signals danger. The vector then unleashes its beneficial genetic instructions. This controlled release mechanism represents a significant step towards minimizing potential long-term effects. Another intriguing development is the exploration of non-viral delivery systems. While viral vectors are incredibly effective, concerns remain regarding immune responses and the potential for insertional mutagenesis. The risk of the vector inserting itself into a location in the genome that disrupts an important gene. Non-viral approaches, such as lipid nanoparticles and exosomes, offer a gentler alternative. Lipid nanoparticles, for example, are tiny bubbles made of fat molecules that can encapsulate and protect genetic material, allowing them to be delivered directly into cells. Exosomes, naturally occurring vesicles secreted by cells, are also being investigated as vehicles for gene delivery, leveraging their inherent ability to interact with and enter cells. These non-viral methods are still in relatively early stages of development compared to viral vectors, but they hold considerable potential for reducing immune responses and improving safety. However, the most exciting developments are emerging from the concept of combination therapies. The understanding of complex diseases, particularly those like cancer and neurodegenerative disorders, is shifting towards the recognition that a single gene modification is rarely sufficient for a lasting cure. Instead, researchers are investigating ways to combine gene therapy with other treatment modalities. For example, in cancer gene therapy, a patient might receive a gene that enhances their immune response, alongside a gene that directly targets and destroys cancer cells. This synergistic approach, often referred to as gene-drug co-therapy, can dramatically improve treatment outcomes. In neurological disorders, combining gene therapy with immunomodulatory treatments is another area of intense investigation. Some neurological diseases are characterized by an overactive immune response that contributes to the progression of the illness. Gene therapy could be used to silence the genes responsible for this overreaction, while simultaneously boosting the immune system's ability to fight off pathogens. Another exciting area is the use of gene therapy to deliver factors that promote neuronal repair and regeneration, combined with drugs that protect neurons from further damage. The goal isn't simply to treat the symptoms of a disease, but to fundamentally alter the underlying biological processes. Furthermore, researchers are beginning to explore cellular reprogramming alongside gene therapy. This involves modifying the cells themselves, not just introducing a new gene, but fundamentally altering their identity. For example, in conditions like Parkinson's disease, scientists are exploring ways to use gene therapy to convert non-functioning glial cells into dopamine-producing neurons. This represents a truly revolutionary approach, transforming the patient's own cells into the missing part of the system. The future of gene therapy isn't just about delivering a single gene. It's about orchestrating a complex interplay of biological events. Advanced vector technologies, coupled with these innovative combination therapies, are paving the way for treatments that are far more precise, effective, and durable than anything we've seen before. The continued advancements in this field promise a future where genetic diseases are no longer considered incurable, but rather treatable conditions. Ongoing research, coupled with careful monitoring and long-term follow-up, will be crucial to ensure that these powerful tools are used safely and effectively, ultimately transforming the lives of countless individuals. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Gene therapy whispers of healing, of potential, now fading into the quiet of your own mind. Let your thoughts drift, calm and unhurried, carried on the gentle currents of sleep.